Hello, scholars. So far too often we see the images of a DNA structure and we start right here. I want to take us back a little bit so that we can really understand the true nature of what DNA is and how it is structured. So we might be familiar with the cell. This right here is a human cell. And inside of the human cell, the most important portion of it, arguably, would be the nucleus. And inside of the nucleus, right here on number one, is this thing called the nucleolus. And what the nucleolus looks like really close up is like this purple circular structure right here. And inside of that structure, what you have are a bunch of these little X like things called chromosomes. We have 46 of them as humans, 46 different types of chromosomes. And so those chromosomes are like wadded up and tangled up and in a ball called the nucleolus, right? And so in there, we end up seeing these little X figures again called the chromosomes. And then as that chromosomes, as we zoom in, we'll notice that these little chromosomes are tied up in like strings or balls of something. And so they string and they kind of wind around themselves in like a coil. And as the coil unravels, you'll see these little balls called histones, where if you go deep, deep, deep into the coils that the histones are wrapped around, you will see this DNA. And so DNA is with this double helix that most of you are familiar with from maybe your biology course or just now taking it this year. You might be able to see this structure in your classrooms. And so if we go really close up, you'll see that this is a ladder. So it's two ladders, or two sides of the ladder, basically here, one side over here and one side over here is one ladder, and it's twisted. And so it's kind of like you take your hands and you twist you take the top of the ladder and you put your hands up there and you twist it, and you take the bottom of the ladder, you put your hands up there and twist it, and it ends up like this structure. But unraveled or untwisted, you will see that it has these little things called base pairs. So this is adenosine, and then so you have the T complement and cytosine, guanosine. So you haven't learned all of these base pairs yet, and it's not important yet for this particular course just yet for you to know these base pair names. So it's okay. But what I wanted you to see is how we start from the cell, and inside of the cell is your DNA, but you can see how it's actually like a Russian doll, right? So you have an egg, or a Russian egg, you have an egg inside of an egg inside of an egg inside of an egg inside of an egg, and that is what we're actually studying today.